Welcome to our bedtime journey, the place where we wander calmly through the wonders of science, where curiosity can drift like clouds, and your thoughts can soften into rest. Tonight, we are going to travel inward, beyond what the eye can see, into the quiet, hidden world of molecular biology. This is the study of life at its smallest scales, the molecules that breathe and sustain everything we are. So, I invite you now to let yourself relax. Take a deep, slow breath in and gently breathe out. Feel the heaviness of your body settling into the bed or chair or wherever you rest. And imagine, just for now, that you are shrinking, smaller and smaller, smaller than a grain of sand, smaller than a dust particle, smaller than the width of a single hair, until you are small enough to wander through a cell, to float among the molecules that hum quietly inside us all. DNA is the library of life. Now that we are small enough, let us begin with the foundation of biology. The molecule called DNA. Inside nearly every one of your cells, there is a nucleus, a tiny chamber where DNA is stored. Think of it as a vast library, a huge collection of volumes, written not in ink, but in code. When we imagine DNA, we often see the double helix, two twisting strands, coiled like a spiral staircase. It is delicate and yet strong, elegant and yet practical, a shape that whispers life. The helix is written in 4 letters, A, T, C, G. Adenine, thymine, cytosine, guanine. Each one is a base, a building block of genetic information. They pair together in a beautiful dance. A with T, C with G. Always faithful, always consistent. Imagine them now, stretching out in pairs, millions upon millions, like beads on a string. Each pair is like a gentle whisper of instruction, a note in the song of life. The scale of DNA. If you were to take all the DNA from a single human cell and stretch it out end to end, it would reach about 2 meters in length. And yet, it fits into a nucleus smaller than the width of a pinprick. How does it fit? By folding, coiling, wrapping around proteins called histones, like threads carefully wound around spools. The cell is a master librarian, storing its encyclopedia with perfect efficiency. And in this encyclopedia lies every recipe for every protein, every enzyme, every structure that makes you, you. DNA replication, copying the library. Now, let's imagine the moment when DNA is copied. This happens whenever a cell divides. The double helix unwinds, unzipping itself like a quiet zipper. An enzyme called helicase moves along, gently separating the 2 strands, as if opening the pages of a book to be read again. Polymerase enters, sliding along the strands, matching bases one by one. A with T, C with G, always precise. It is as if the cell whispers, we must remember, we must preserve this story. And so, the DNA makes a copy of itself. Faithful, accurate, with only the rarest of mistakes. DNA is a living story. Let us pause here and reflect. DNA is not just a molecule. It is a story, a record of ancestry, a thread of memory, woven through time. From the first spark of life, billions of years ago, all the way to you, lying here tonight. Your DNA carries echoes of ancient oceans, of ancestors who breathed, walked, loved, long before you were born. This story is still being written. Each cell, each moment, each replication is a continuation of that quiet history. So rest here for a moment, breathing gently, imagining the helix turning slowly, a ladder of light spiraling endlessly in the dark. RNA and the message. We have seen now how DNA is the great library of life. Silent, stable, storing instructions. But DNA rarely leaves the nucleus. It's too precious, too heavy with information. Instead, the cell uses a messenger, a lighter copy, a traveling note. This is RNA, ribonucleic acid. The birth of RNA and transcription. Inside the nucleus, a process called transcription begins. An enzyme called RNA polymerase moves along the DNA. It reads one strand, base by base, and creates a matching chain of RNA. DNA says A. RNA answers U. DNA says C. RNA answers G. One base after another, a new message is written. This is not the full library. It is a single recipe, a fragment, carried outward. We call this copy messenger RNA, or mRNA. The structure of RNA. How is RNA different from DNA? First, RNA only has one strand, not two. It is like a ribbon instead of a ladder. Second, one base is different. Where DNA uses thymine, or T, RNA uses uracil, U. So, adenine pairs with uracil. Cytosine still pairs with guanine. This small change makes RNA less stable than DNA. It is temporary. A message meant to be used and then discarded. RNA processing. In the complex cells like ours, the first copy of RNA is not ready yet. It must be processed. Some sections, called introns, are removed. Other sections, exons, are stitched together. The ends are modified too. A cap is placed on one end, and a long tail of adenine bases is added to the other. This protects the RNA. It tells the cell, this is ready to travel. There is a journey out. Now imagine this finished messenger RNA slipping out of the nucleus through tiny pores in the nuclear envelope. It enters the cytoplasm, a bustling city of organelles, molecules, and movement. Here, the message will be read, translated into a protein. The genetic code. Before we continue, we must understand how the message is written. The letters of RNA are read in groups of 3. Each group of 3 bases is called a codon. For example, AUG. This codon means start. It tells the ribosome where to begin. Other codons specify amino acids. For instance, UUU codes for phenylalanine. GGC codes for glycine. There are 64 possible codons, and they all cover 20 amino acids. This is the genetic code, a universal language shared by almost all living things. Translation: the ribosome. Now, the messenger RNA finds a ribosome. A ribosome is a molecular machine, part protein, part RNA. It grips the messenger and begins to read it, one codon at a time. For each codon, another molecule arrives. This is transfer RNA or tRNA. Each tRNA carries an amino acid. One end matches the codon, the other end carries the amino acid. When the codon and the tRNA match, the ribosome locks the amino acid in place, and then it adds the amino acid to a growing chain. Building a protein. Codon after codon, amino acid after amino acid, the chain grows. Think of beads being strung onto a necklace, or bricks laid into a wall. At last, the ribosome reaches a stop codon. The chain is released. It folds, bends, twists into a three-dimensional protein, and the message has been fulfilled. The role of RNA in life. RNA is more than just a messenger. Some RNAs form part of the ribosome itself. Some act as tRNAs. Some regulate genes, turning them on or off at the right moment. And in ancient history, RNA may have been the very first molecule of life. A world where RNA both stored information and carried out reactions. The RNA world hypothesis. Even today, viruses like influenza or coronaviruses use RNA as their genetic material. So this fragile strand, this fleeting note, is central to all of biology. Let's pause for reflection and reflect on what we have learned. DNA stores information. RNA carries it outward. Ribosomes translate it into proteins. It is like a cycle of language. A library writes the book. A messenger delivers it. A translator speaks it aloud. And finally, a worker builds from these words. This cycle, called the central dogma of molecular biology, is the foundation of life. And as you rest here, listening to these quiet details, your own cells are doing this right now, silently, endlessly. Proteins are the builders of life. So far, we have seen how DNA holds the information, how RNA carries the message, and now we arrive at the result, the workers, the builders, the molecules that do most of the actual tasks of life. These are the proteins. What are proteins? Proteins are long chains of amino acids. There are 20 different amino acids used by life on Earth. Each has its own character. Some are small, like glycine. Some are bulky, like tryptophan. Some carry a positive charge, others negative. Some are oily, repelling water. Others are attracted to water. By linking these amino acids in different orders, cells create proteins with unique properties. It is like an alphabet of 20 letters. From these letters, the cell writes words, and those words become tools. Proteins fold. When a protein chain is first made at the ribosome, it is just a simple strand, but immediately it begins to fold. The chemical nature of each amino acid causes the chain to twist, to loop, to bend. Some sections form spirals called alpha helices, or, in singular terms, an alpha helix. Others form flat sheets called beta sheets, or beta-pleated sheets. And these pieces fold further into complex three-dimensional shapes. The final form of a protein is not random. It is dictated by the sequence itself. The chain knows how to fold. and the shape determines the function. Imagine tiny origami folding itself perfectly in a fraction of a second. Proteins have functions. What do proteins do? Almost everything. Some are enzymes. These speed up chemical reactions. Without enzymes, life would be impossibly slow. Others are structural proteins. Collagen, for example, gives strength to your skin and bones. Keratin builds your hair and nails. Some proteins are transporters. Hemoglobin, for example, carries oxygen into your blood. Membrane channels move ions across cells. Still others are signaling proteins. Hormones like insulin are signaling proteins. They tell cells when to store sugar, when to use energy, and when to grow. Proteins are also defenders. Antibodies are proteins, seeking out invaders, marking them for destruction. Wherever you look in biology, you find an assembly and a variety of diverse proteins at work. There is a beauty to specificity. What makes proteins remarkable is their specificity. An enzyme will recognize one particular molecule and bind to it as if it were made for it. This is sometimes called the lock and key model. The protein is the lock, the molecule the key. Only the right shape will fit. This specificity gives life its precision. It allows millions of reactions to happen at once, without confusion. There are chaperones helping proteins fold. Sometimes proteins need help folding correctly. The cell provides special helper proteins called chaperones. These do not become part of the final structure. They simply guide the folding, preventing errors, ensuring the protein finds its right shape. Think of them as teachers, gently correcting the folds in the origami until the shape is perfect. But what about misfolded proteins? What happens when folding goes wrong? Trouble can follow. Misfolded proteins can clump together and the forming aggregates. These are linked to diseases like Alzheimer's, Parkinson's, and Huntington's disease. Even the smallest mistake in folding can lead to large consequences, and yet, most of the time, cells succeed. Day after day, they fold proteins with astonishing accuracy. The large scale of protein work. Consider this. A human cell may contain over 10 billion proteins, each one with a role, each one working independently or collaboratively. Some last for only minutes before being recycled. Others remain for days or weeks. Together, they maintain the balance of life. Let's pause for another reflection. So now, as you rest, think of the proteins at work inside you. Enzymes are breaking down food into energy. Structural proteins holding your tissues in place. Transport proteins moving oxygen into your cells. Signaling proteins carrying messages. All of this happening quietly, effortlessly, without you ever noticing. The builders of life, always working and always present. Life has energy and flow. We have explored DNA, the keeper of the code. We have followed RNA, the faithful messenger. We have seen proteins, the builders and workers of life. But there's one question left. How are they powered? What gives cells the energy to carry out their endless tasks? The answer lies in a small molecule known as ATP. ATP is the energy currency. ATP stands for adenosine triphosphate. It is the universal energy currency of the cell. Imagine a rechargeable coin. Every time a cell needs energy, it spends a coin of ATP. When the task is done, the coin can be recharged and used again. ATP is simple in structure. A sugar, a base, and 3 phosphate groups. The bond between these phosphate groups stores energy. When one phosphate is cut loose, energy gets released. ATP becomes ADP, adenosine diphosphate, and later it can be recharged. This constant cycle is like the quiet heartbeat of the cell. But where does ATP come from, and what is metabolism? How does the cell make ATP? The answer is metabolism. The sum of all the chemical reactions in the body. One major source is glucose, a simple sugar. When you eat bread, rice, or fruit, glucose enters your bloodstream. Cells absorb it and break down to harvest the energy of glucose. The process begins with glycolysis, a set of reactions that split glucose in half. This yields a little ATP, but the real payoff comes later. The halves of glucose are fed into another cycle known as the Krebs cycle or the citric acid cycle. Here, high energy electrons are harvested. These electrons are passed to the electron transport chain, or ETC, a series of proteins in the mitochondria. As electrons flow down the chain, protons are pumped, gradients are formed, and finally, ATP is made in great quantities. Mitochondria, as you may have heard before, is the powerhouse of the cell. This brings us to what mitochondria really does. These organelles are tiny, but inside them lies an elegant system for making energy. A power plant. Mitochondria have 2 membranes, an outer one, smooth and simple, an inner one, folded into ridges called cristae. Along these folds, the electron transport chain is embedded. Here, oxygen accepts electrons at the very end, combining with protons to form water. This process, oxidative phosphorylation, reduces the majority of ATP in your body. Every breath you take delivers oxygen to this chain. Every heartbeat delivers glucose, and mitochondria unite them to create the energy of life. Energy needs balance. Cells are careful with their energy. Too little ATP, and processes stop. Too much, and resources are wasted. Sensors monitor the energy state, activating pathways as needed. When ATP is low, cells burn more glucose or fat. When ATP is abundant, cells store the surplus. This balance keeps life steady, not too fast, not too slow. There are other fuels as well. While glucose is common, cells can use other fuels as well, like fatty acids, which yield even more energy, broken down through beta oxidation. Amino acids can be used too, though usually as a last resort, since they are needed for proteins first. No matter the source, the end goal is the same: to make ATP. Now let's talk about the flow of energy in life. Energy is not created, it is transformed. Plants capture sunlight, turning it into sugars. Animals eat the plants, breaking sugars into ATP. Predators eat other animals, passing energy along further. And at every step, some energy is lost as heat. The warmth of your body, the heat of a campfire, the glow of life itself, all are reminders of this flow. So let's reflect once more as you lie here right now. Your cells are quietly making ATP. Each heartbeat and each breath fuels the mitochondria, and the mitochondria fuel the work of life. The proteins folding, the signals passing, the repairs being made. All of it is powered by tiny sparks, coins of energy, spent and renewed, over and over again. It's a rhythm older than humanity, older than animals, older even than plants. It is the rhythm of life itself. Cells have membranes, and in those membranes, there are transport systems. So far, we have wandered inside the cell. We have seen the DNA, the RNA, the proteins, and the flow of energy. But the cell itself is not an island. It has a border, a delicate, shimmering boundary that separates inside from outside. This is the cell membrane. This membrane has a lipid bilayer. The cell membrane is made of lipids. Lipids are fatty molecules, each with two parts, a head that loves water and a tail that fears it. When placed in water, these lipids arrange themselves in a double layer, heads facing outward, tails tucked inwards. This is the lipid bilayer, a thin, flexible sheet, only a few nanometers thick, and yet this fragile sheet is strong enough to hold the contents of life. Let's talk about selective permeability. The membrane is not just a wall, it is selective. It decides what can pass and what must stay out. Small molecules like oxygen and carbon dioxide can slip through freely, but larger or charged molecules, they require help. This control maintains the cell's balance. It keeps the inner world stable, even when the outer world changes. How do we transport proteins? To manage this traffic, the cell uses transport proteins. Some are channels. They form pores, allowing ions like sodium or potassium to flow in or out. Others are pumps. They use energy, often ATP, to move molecules against their gradient. Still, others are carriers, gripping a molecule on one side and releasing it on the other. Each protein is specific. Each plays a role in maintaining life. The sodium-potassium pump. One famous example is the sodium-potassium pump. The pump moves 3 sodium ions out of the cell and brings 2 potassium ions in. It does this over and over, using ATP each time. The result is an electrical gradient, a difference in charge across the membrane. This gradient powers nerve impulses, muscle contractions, and countless other processes. Even as you lie here now, billions of pumps are quietly at work, maintaining that balance. Membranes are fluid. The membrane is not rigid, it has fluidity. Lipids drift, proteins slide. It is sometimes called the fluid mosaic model. The fluidity allows the cell to change shape, to engulf particles, to fuse with other membranes. Imagine a calm, rippling surface of water, with boats drifting, shifting gently with the currents. That is the membrane. Let's think about the cell membrane as the guardian of life. It holds the inside in and keeps the outside out. It is both a shield and a gate. Through it, nutrients enter, wastes leave, signals arrive. It is thin, flexible, almost delicate, and yet, without it, once again, life would not exist. How do cells communicate? We have seen how the cell protects itself with membranes, but cells do not live in silence. They need to sense, to respond, to communicate. This constant exchange of signals is called cell signaling. Without it, cells would be blind and deaf. They could not respond to their environment, nor coordinate with their neighbors. There is a language of signals. Signals can take many forms. Some are small molecules, like hormones. Insulin, for example, tells cells to take in glucose. Others are gases, like nitric oxide, which relaxes blood vessels. Some signals are proteins or peptides, traveling through the bloodstream. and subsignals are simply ions, like calcium, flooding into a cell to trigger muscle contraction. Each signal carries a message. Each message must be received. The receptors are the listeners. For a signal to have meaning, a cell needs a receptor. Receptors are proteins that sit in the membrane or inside the cell. They are tuned to recognize specific signals. When a signal binds, the receptor changes shape. This change is the start of the message inside. There are different kinds of receptors. Some are ion channel receptors. When the signal binds, the channel opens, letting ions flow. Some are G protein coupled receptors. Don't be intimidated by the name. These are versatile, activating cascades of proteins inside the cell. Others are enzyme-linked receptors. When activated, they trigger chemical reactions directly. And some receptors, like those for steroid hormones, sit inside the cell, waiting for the signal to slip through the membrane. Cascades are often the amplifiers. Often, the binding of a signal is just the beginning. Inside the cell, a chain of events follows. This is called a signaling cascade. One protein activates the next, which activates another, and another still. Like a line of falling dominoes, a single signal can lead to a vast response. This amplification ensures that even a tiny signal is never lost. Second messengers. Many cascades rely on small molecules called second messengers. One famous example is cyclic AMP, or CAMP. It spreads through the cytoplasm, activating proteins. Another is calcium ions, which surge suddenly, like waves washing through the cell. These messengers carry the signal forward, ensuring that they reach the right targets. Cells have specific pathways. One famous cascade is the MAP kinase pathway. It begins with the growth factor binding to a receptor. This activates a small protein called RAS, or RAS. RAS triggers a series of kinases, enzymes that add phosphate groups to proteins. The signal flows from one kinase to another until it reaches the nucleus. Here, it activates genes that drive cell growth and division. Another example is the insulin signaling pathway. When insulin binds to its receptor, a chain of events leads to glucose transporters being moved to the membrane. The cell can now take in sugar from the blood. These pathways are precise, yet flexible. They allow cells to respond to the world with balance and control. Let's talk about the communication between cells. Signals can act in different ranges. Some signals act locally, affecting only nearby cells. This is called paracrine signaling. Others act on the same cell that released them. This is autocrine signaling. Some signals travel long distances through the bloodstream. This is endocrine signaling, used by hormones. And some require direct contact, cell to cell. This is juxtacrine signaling, just like the word juxtaposition, like a whisper between neighbors. When signaling goes wrong. When signaling is precise, cells thrive. But when it goes wrong, disease can result. If signals that drive growth never turn off, cells may divide uncontrollably. This is cancer. If insulin signals are ignored, cells fail to take in glucose. This is diabetes. If immune signals misfire, the body may attack itself. This is autoimmunity. So, signaling must be balanced, carefully tuned, like an orchestra. As you rest now, remember that your cells are listening. Every moment, receptors feel for signals, cascades flow. Messages are amplified. Like a quiet city at night, lights flickering, voices murmuring. Your body is alive with whispers. Signals carried through your tissues, coordinating growth, healing wounds, keeping balance. It's a conversation older than language, older than thought, older than animals. Let's talk about the cell cycle and cell division. In molecular biology, one of the most fundamental processes is the cell cycle. It is the ordered sequence of events that allows cells to grow, copy their DNA, and divide into two daughter cells. This cycle is essential for development, tissue repair, and even day-to-day maintenance of the human body. Right now, millions of your cells are going through this cycle, replacing old cells, healing tiny injuries, and keeping you alive. Let's walk through it carefully, step by step. Stages of the cell cycle. The cell cycle is traditionally divided into 4 main phases. The first one is G1 phase, or gap 1. The cell grows, it produces proteins, and prepares for DNA replication. It's a period of high metabolic activity. Cells in G1 are busy making enzymes and organelles. Then there's S phase, or synthesis phase. This is when DNA replication occurs. Every chromosome is duplicated, resulting in 2 identical copies called sister chromatids. DNA replication is precise, but it also has built-in proofreading mechanisms just to make sure that we can correct mistakes. This is followed by G2 phase, or gap 2. The cell continues to grow and makes final preparations for division. It checks the replicated DNA for errors and repairs them if possible. Organelles like mitochondria are duplicated. Finally, we have the 4th phase, or otherwise known as the M phase, for mitosis. The cell divides its copied DNA and cytoplasm to create 2 daughter cells. This phase includes mitosis and cytokinesis, which we will explore in detail. Between these phases, there are checkpoints, control systems that ensure that the cycle only continues if the conditions are correct. Interphase, or the preparation. In the first 3 phases, G1, synthesis, and G2, are collectively known as interphase. Interphase is not a resting stage, despite what the name suggests. It's a period of intense activity during which the cell is preparing for the crucial act of division. Most of a cell's life is actually spent in interphase, not in mitosis. Mitosis is when we have the division of the nucleus. Mitosis itself is divided into 5 substages: prophase, prometaphase, metaphase, anaphase, and telophase. An easy way to remember this is by PPMAT. Prophase is when the chromatin condenses into visible chromosomes. Each chromosome consists of 2 sister chromatids, joined at a centromere. The mitotic spindle, made of microtubules, begins to form. The nuclear envelope starts to break down. In prometaphase, the nuclear envelope disappears almost completely. Spindle fibers attach to chromosomes at regions called kinetochores. Chromosomes begin moving toward the center of the cell. Now in metaphase, chromosomes line up along the middle of the cell, or the metaphase plate. This alignment ensures that the new cell will receive 1 copy of every chromosome. During anaphase, sister chromatids are pulled apart by spindle fibers. They move to opposite poles of the cell. This will guarantee genetic equality between daughter cells. Finally, telophase. The chromosomes reach the poles and begin to decondense back into chromatin. Nuclear envelopes reform around each set of DNA. The spindle disassembles. Following mitosis is cytokinesis, when the cytoplasm divides. In animal cells, a contractile ring pinches the cell into two. In plant cells, a cell plate forms, building a new wall between the daughter cells, since plant cells have cell walls instead of cytoplasms. Checkpoints and regulation. The cell cycle is tightly regulated. At each checkpoint, proteins check for DNA damage, adequate resources, and proper alignment. The G1 checkpoint ensures that the environment is favorable. The G2 checkpoint verifies that the DNA replication is complete and error-free. The M checkpoint during metaphase confirms that all chromosomes are attached to spindle fibers. Proteins called cyclins and cyclin-dependent kinases will control progression through the cycle. Their levels rise and fall, acting like timers. If problems are detected, the cycle can pause. If damage is irreparable, the cell may undergo apoptosis, otherwise known as programmed cell death, to protect the organism. Why is this important? Why do we need to know about the cell cycle? The cell cycle is important for growth and development, from a single fertilized egg or zygote to trillions of cells. For repair, healing cuts, regenerating tissues, replacing dead cells, or maintenance, constantly renewing skin, blood, and gut lining. Errors in cell regulation and the cycle can be dangerous. If checkpoints fail, cells may divide uncontrollably. This is one of the hallmarks of a tumor, which can lead into cancer. Thus, the cell cycle represents both life's renewal and its vulnerabilities. As you listen to this, your bone marrow is producing new blood cells. Your skin is creating new cells to replace the old. Your intestines are refreshing their lining. But don't fear about errors, because your body is doing this at such a precise level. This quiet cycle of growth and renewal continues through your life. It is structured, orderly, and carefully controlled. And it is happening right now within you. We have walked through the library of DNA. We have followed the messengers of RNA. We have seen the builders, the proteins. We have watched energy flow through mitochondria. We have touched the membranes and listened to the whispers of cell signaling. We have seen the rhythm of division. Now let us step back and simply listen to the harmony of it all. The orchestra of the cell. Imagine your body as an orchestra. DNA is the composer, holding every note of the score. RNA is the sheet music carried to each musician. Proteins are the players, each with an instrument, each performing their own part. ATP is the rhythm, the steady beat of the drums, keeping time for every note. The membranes are the stage, holding the orchestra together. The signals are the conductor's gestures, guiding the performance, ensuring harmony. And the cycle of cell division is the quiet renewal of the symphony itself, always beginning again. Now, let us slowly zoom out from one protein folding in silence, to one ribosome reading its message, to one cell humming with activity. Now a tissue, millions of cells working side by side. Then an organ, a heart that beats, a brain that thinks, a lung that breathes. And finally, you, the person who holds all of this within. Every heartbeat, every breath is supported by the molecules we have explored. As you lie here tonight, your cells are not sleeping. They are still dividing, still signaling, still making proteins, still weaving the story of your life. They do not tire, they do not forget, and they do not rest. And because of this, you can rest. Your body, in its molecular harmony, carries you gently into tomorrow. If you let your imagination wander further, you might see that this harmony is not just in you. The same DNA code is written in all living things, in the trees outside your window, in the birds that fly at dawn, in the tiny microbes that live unseen. Life, in all its diversity, shares the same molecular dance. And if we go further still, the atoms that makes these molecules were forged in stars, billions of years ago. In the heat of exploding suns, you are not only made of cells, you are made of stardust. So let this be your lullaby tonight. The DNA whispering its code, the RNA carrying its notes, the proteins folding their instruments, the mitochondria pulsing like lanterns, the membranes rippling like calm water, the signals weaving like voices in a choir, the cell cycle renewing the performance over and over again. You are the symphony. You are the harmony. You are the music of life itself. And now, as the music of the molecules continue without you, allow yourself to drift. Let your breath grow slower. Let your body grow heavier. And let your thoughts soften like mist dissolving into night. Trust the harmony within you. Trust the orchestra that never stops. And rest. Good night.